My name is Jackson Shelley, and today is May 4th, 2023. So this is my presentation for the Diseases of the Cell Project on hemolytic anemia. Hemolytic anemia is a condition characterized by premature destruction of red blood cells, which can lead to a variety of metabolic and biochemical imbalances in the body. Normally, red blood cells are produced in the bone marrow and they circulate in the bloodstream for about 120 days before being broken down and recycled by the spleen and liver. In hemolytic anemia, this process is disrupted, leading to a shortage of red blood cells in the body. Here is a visualization of hemolytic anemia. So there's a normal red blood cell versus one that has hemolytic anemia, and as you can see, they are broken down partially, and so it does cause problems for functions in the body. So there are two types of hemolytic anemia: inherited, which is caused by genetic mutations that affect the production or structure of red blood cells. Examples of this include sickle cell anemia, thalassemia, hereditary spherocytosis, elliptocytosis, and glucose 6-phosphate dehydrogenase deficiency. And there's also acquired, and these are caused by factors outside of genetics, such as infections, drugs, autoimmune disorders, or certain medical practices. Metabolic pathways that are damaged by hemolytic anemia include there's an increased breakdown of heme. So oxygenase is the enzyme that catalyzes the breakdown of heme, and it is upregulated during hemolytic anemia. There's also the pentose phosphate pathway, so NADPH and other metabolites, which are necessary for this pathway, are depleted as a result of hemolytic anemia, which would therefore downregulate pentose phosphate pathway. In addition to these pathways, hemolytic anemia may also lead to alterations in other metabolic pathways and enzymes, including those involved in producing and degrading various amino acids, lipids, and carbohydrates. Functions of the cell that are impacted by hemolytic anemia include reduced oxygen delivery. So a red blood cell's primary job is to carry oxygen from the lungs to the rest of the body, and with the breakdown of these red blood cells, that would disrupt the process. There's a disruption of the electrolyte balance. So red blood cells are responsible for upregulating levels of various electrolytes and metabolites in the blood, such as potassium, sodium, and lactate. Further, they have increased oxidative stress, so due to the red blood cells storing a lot of heme and other molecules that produce reactive oxygen species, they're especially vulnerable to oxidative harm. Excuse me. Is hemolytic anemia influenced by epigenetic factors? So there are two categories of epigenetic modifications that affect how genes involved in the production and operation of red blood cells are expressed, and those are variations in DNA methylation and histone modification. So, for instance, modifications in DNA methylation of a beta globin's gene promoter region, which codes for a component of hemoglobin, have been related to chances in gene expression that aid in the onset of sickle cell anemia. Additionally, it has also been demonstrated that environmental factors like stress or exposure to chemicals can change the epigenetic modifications that will disrupt the synthesis of red blood cells and help cause hemolytic anemia. So the pattern of inheritance for hemolytic anemia, so both sickle cell anemia and thalassemia are autosomal recessive diseases, which means that the two copies of the defective gene, one from each parent of course, must be inherited by a person in order to have the disease. Now, hereditary spherocytosis and hereditary elliptocytosis are inherited in an autosomal dominant way, which means that one copy of the defective gene from one parent is all that is required for them to have hemolytic anemia. Now, a glucose 6-phosphate disorder deficiency is an X-linked disease, which means that the gene is found on the X chromosome. Since males are the only ones with an X chromosome, they will get the disorder when they inherit the mutant gene. Mutations in genes that encode for, genes involved in hemolytic anemia. Mutations in genes that encode for hemoglobin proteins such as HBB and HbA1/2, HbA2, is one of the most prevalent hereditary causes of hemolytic anemia. The KLF1, the croup-like factor 1 gene, also known as the EKLF, erythroid croup-like factor, is a transcription factor that plays a critical role in the regulation of erythropoiesis, which is the process by which red blood cells are produced. Genes encoding for membrane proteins are also thought to contribute to hemolytic anemia. A variety of hemolytic anemias can be caused by mutations in these genes, which are essential in maintaining the structure and functionality of red blood cells. Two examples of genes encoding for membranes are the ANK1, which encodes for a protein that aids in anchoring the membrane to the cytoskeleton, and the SLC4A1, which codes for proteins involved in the exchange of ions across the red blood cell membrane. So, is hemolytic anemia caused by mutations? Hemolytic anemia can be caused by genetic mutations that affect the structure and function of proteins involved in the red blood cell life cycle, such as hemoglobin, enzymes, and membrane transporters. The type of mutation involved in hemolytic anemia can vary depending on the underlying genetic defect. So, for example, sickle cell anemia is caused by a point mutation in the HBB gene that leads to the production of abnormal hemoglobin, while hereditary spherocytosis is caused by mutations in the genes ANK1, SPTB, or SLC4A1 that encode for proteins involved in the red blood cell's membrane structure and stability. Diagnosis methods for hemolytic anemia include blood tests, so a CBC or complete blood count is typically the first step in diagnosing anemia. The test measures the number of red blood cells, white blood cells, platelets in the blood, as well as the levels of hemoglobin and hematocrit. Have normal results which suggest hemolytic anemia. A blood smear. So a blood smear involves examining a sample of blood under the microscope to look for abnormalities in the shape, size, and color of the red blood cells, and this can help to identify the underlying cause of hemolytic anemia. The Coombs test. So this test detects the presence of antibodies that attack and destroy red blood cells, which can help to diagnose autoimmune hemolytic anemia. Hemoglobin electrophoresis. So this test is used to detect abnormal hemoglobin, which can cause hemolytic anemia in conditions like sickle cell anemia and thalassemia. There is also a bone marrow biopsy. So in some cases, a bone marrow biopsy may be performed to examine the production of red blood cells in the bone marrow and identify any abnormalities. So is a specialist needed for hemolytic anemia? If hemolytic anemia is suspected due to an infection or medication, a primary care physician may order initial results or treatment. Now, if the anemia is suspected to be caused by a genetic disorder or autoimmune disease, a referral to a hematologist or other specialist may be necessary for further evaluation and treatment. So, preventative technologies for hemolytic anemia. So in some cases, such as the inherited forms of hemolytic anemia, like sickle cell anemia or thalassemia, genetic counseling and testing can help to identify the carriers of this condition and allow for informed family planning decisions. In other cases, prevention of hemolytic anemia may involve avoiding exposure to certain medications or toxins that cause the condition. For example, some medications like antibiotics, chemotherapy drugs, and non-steroidal anti-inflammatory drugs, or NSAIDs, can trigger autoimmune hemolytic anemia in susceptible individuals. So there are a couple treatments for hemolytic anemia. So medications, depending on the underlying cause of hemolytic anemia, medications may be used to suppress the immune system, reduce inflammation, or prevent the destruction of red blood cells. Examples of medications that may be used to treat hemolytic anemia include corticosteroids, immunosuppressants, and intravenous immunoglobulin. There's also blood transfusions. So in several cases of hemolytic anemia, blood transfusions will be necessary to replace the destroyed red blood cells and restore oxygen carry capacity. Transfusions may be needed on a regular basis depending on the severity of the anemia. Plasmapheresis. So this is a procedure in which the plasma, the liquid component of the blood, is removed from the blood using a centrifuge and replaced with other fluids. The plasmapheresis may also be used to remove antibodies that are attacking and destroying red blood cells in case of autoimmune hemolytic anemia. There's also a bone marrow transplant. So in certain types of inherited hemolytic anemia, a bone marrow transplant may be considered to replace the faulty bone marrow with healthy donor cells. Immunoglobulin therapy, so using large doses of immunoglobulin to suppress the immune system and lessen the destruction of red blood cells, is the main goal of this treatment. So how do allied health professionals help with those patients with hemolytic anemia? So dietitians can help patients create a well-balanced diet, high in nutrients such as iron and folic acid, which are necessary for red blood cell formation. Dialysis technician, so patients with hemolytic anemia may often require dialysis to manage their condition. Physical therapist, so hemolytic anemia patients may experience weakness, fatigue, or muscle pain, which will limit the degree of their physical exercise. Physical therapists can collaborate with their patients to create a secure exercise program that enhances strength, flexibility, and management. So the state of research on hemolytic anemia. So there's active research being conducted on hemolytic anemia with the aim of improving our understanding of the disease and developing new, more effective treatment. This research includes studies to identify new genetic mutations associated with hemolytic anemia, investigate the novel therapies such as gene therapy or immunomodularity agents, and explore the use of stem cell transplantation in the treatment of inherited hemolytic anemia. Here are my primary sources, and here are my secondary sources. Thank you.